A recent increase in toxic red tide outbreaks caused by blooms of saltwater dinoflagellates created dead zones in the ocean by depleting oxygen levels. Researchers are not sure exactly what is causing the increased outbreak of red tides and algal blooms, but sewage, atmospheric deposition, groundwater flow, and agricultural runoff have greatly increased nutrient levels along the east and west coasts, which have been linked to increased algal blooms. Researchers hypothesize that agricultural runoff containing pesticides has increased levels of nutrients like nitrogen, which facilitate quick algal growth. We conducted this experiment to determine the effect that increasing nitrogen levels has on the marine dinoflagellates Amphidinium, Gymnodium, and Pluricentrum. Based on research conducted by Anderson, we hypothesized that high nitrate levels increase dinoflagellate growth in salt water. Procedures for making the algae media were followed according to the Carolina Biological Supply Company's Culturing Algae Manual. The Erdschreiber medium was created by combining artificial seawater, a natural soil water supernatant, and a salt solution. Each element was autoclaved separately and then combined. 7.5 milliliters of saltwater algae media and 0.375 microliters of the dinoflagellate mixture were added to 5 test tubes. Each of the 5 test tubes contained different nitrogen levels of either 0, 1, 10, 30, or 100 micromoles of nitrate. For reference, nitrogen levels of 0.2 to 30 micromoles are usually found in natural ocean conditions, according to the Encyclopedia of Life. 3 trials were set up and conducted simultaneously. Tubes were then stored in an incubator at 22 degrees Celsius. Thermodimetry readings were taken every 12 hours for 12 days. The photospectrometer was blanked using a tube with only algae media. Each tube was inverted to allow even distribution of algae cells, and absorbance readings were taken. Cell counts were taken using a hemocytometer. Cell counts could only be performed for the first 6 days in order to avoid removing too much algae media for the photospectrometer to read. A small amount of iodine was added to kill the cells and make them easier to count. This graph shows the average log of the absorbance readings taken twice a day for 12 days. Each line represents different concentrations of nitrate. The data shows that each tube increased at relatively the same rate, indicating that increasing the concentration of nitrate had no impact on increasing growth rate, supporting our null hypothesis. If our hypothesis were supported, we would have expected larger differences between the algae with 0 micromolar and 100 micromoles of nitrate. Growth rates were calculated for each nitrate concentration, ranging from 0.00053 to 0.00051. This further supports our conclusion that nitrate levels had minimal effects on growth rates. This graph compares day 1 cell counts to day 6. Here we see large differences between varying concentrations of nitrate. However, we are not confident in the accuracy of the cell counts based on the high fluctuations from day to day. This graph shows the average count of cells per milliliter based on different populations with different nitrate levels. Based on the data, there is no correlation between number of cells and increasing the nitrate levels, as all of the populations increase at relatively the same rate. A possible explanation for varying results can be attributed to different people taking cell readings and less experience. Dixon and Surratt performed a similar experiment using a hemocytometer to count algae. They found that increased levels of algae and arginine increased the growth of saltwater algae by 50%, while ammonium proved toxic to the algae. According to our average results for the thermodometry readings, nitrogen had little impact on dinoflagellate growth rates, meaning that nitrogen was not a limiting factor on the dinoflagellates. Davidson et al. Found that phosphorus may be a limiting factor, as the usual ratio of carbon to nitrogen to phosphorus is 106 to 16 to 1. The 100 micromolar nitrogen level had slightly higher growth, which may indicate that if nitrogen levels were increased further, growth may increase with it, but more tests would need to be conducted.